Okay, let's see. Today was going to start talking about genome-wide association studies, GWAS stuff. All right. In the topic of next-gen sequencing, there's some more stuff in this NGS.R script that let's just briefly look at and then move on. So we have most recently talked about, in the context of next-gen sequencing, how to do statistical tests. So if we, say, had RNA counts, RNA-seq data, and we wanted to go through and ask of our thousands and thousands of genes which ones are differentially expressed between some conditions. There's EDGE-R, there's DE-seq2, there's also that lemma thing that I don't really recommend. But DE-seq2 is a good tool for p-values and false discovery rates with next-gen sequencing data. So I'm going beyond that chunk of code, the EDGE-R, DE-seq2, and lemma stuff. And I'll just briefly show you what more is in here. So there is a package on Bioconductor that you can use to do gene ontology enrichment with RNA-seq data, next-gen sequencing data. Remember what Go enrichment is? We talked about it earlier in the semester. You can do that with negative binomial type models appropriate to read count data. You can do that here using the GoSeq package, GoSeq, if you're interested. You can do KEGG enrichment, separate package. There's a blurb in here about methylation analysis. What's that about? I'm not going to go through this in detail, but this epigenetics idea that I've mentioned before is the idea of the activity of your genes being regulated by the DNA itself. So RNA can come in and regulate what happens with certain genes. Proteins can come in and bind to the DNA and regulate how active a gene is, things like that. That's referred to as epigenetics, the regulation of gene expression that does not require changes to the genetic code itself. And one common mechanism of regulation is methylation. Methylation means a methyl group is added to either a C or an A in a DNA sequence. So a methyl group, what is that, CH, what's a methyl group? CH3? Yeah, okay. So if there was a C at a particular location in the DNA, and there's a little carbon with 3 hydrogens sticking off of it floating around, and for some reason comes and binds to, sticks to that C, then methylation has happened at that location. And who cares? You care because that could regulate the expression of the gene or genes that are nearby. Okay, so knowing that methylation is a common genetic regulatory mechanism, suppose we wanted to, in the context of maybe cancer, suppose we wanted to locate in the genome where is methylation happening. So here's some cancer people, here's some healthy people. Let's try to identify, there's methylation at these genes in the cancer people, there's not methylation at those genes in the cancer people. That's the idea. It's just next-gen sequencing, but we are specifically searching for locations where methylation has happened. How would you do that? We're doing sequencing, and all sequencing knows how to do is sequence. Give me A's, C's, T's, and G's. So how can I use that to find locations of methylation? Well, if you remember what I said at one point about transcription factors. They're in the DNA. So here's a strand of DNA, double helix with the A's and C's and T's and G's bound together. Like that. And so that obviously extends well off the screen, millions and millions of these locations. But suppose that's a gene right there. If you zoomed in onto chromosome 4, location base starting at 500,022, these bases constitute a gene, say. A transcription factor is a protein that can come and physically stick to the DNA. And when it does that, it affects the ability to make a copy of this gene and go put it into action as a protein. So it can regulate turn up, turn down, the expression level by binding there. And with next-gen sequencing, we can do something similar to methylation. We can say, this particular transcription factor X, I would like to know in these rabbits, where does that transcription factor bind? Where does it operate in the genome? What we can do with next-gen sequencing, roughly speaking, is get a bunch of copies of transcription factor X, throw them in together with the DNA, let things mingle, and X, after time, should go stick wherever it can. So all the places in the genome where transcription factor X operates, ideally if we let it mingle long enough and we have enough copies of everything, will go stick everywhere it's supposed to stick, that it's designed to stick. So then, what if we did sequencing in such a way that we could separate out the reads that have X sticking on them? So we have this DNA molecule, enormous molecule, and at various places along it, the transcription factor has bound. So if we can break the DNA like we have to do when we do next-gen sequencing, break it all up into tiny pieces, we would have tons of little fragments, and some of them would have the transcription factor bound to them. Some of them would not. So all we would need to do is separate out the reads that have the transcription factor on them. So you can do chemistry in the lab to do that. Only pull out the reads that have a protein sticking on them. And then all you have to do, well, I'd say all you have to do, what they have to do in the lab is kick off the protein. So after we kind of filter out, we would be left only with the reads that have the transcription factor on them. And then we need to kick off the protein and then do the sequencing. So kick off the green protein. Now all you have is just a bunch of reads, and we're ready to do next-gen sequencing as usual. Once you do that, if you can align those reads back to the genome, you can find where the transcription factor bound. So if we were to take that read and align it back to the genome, that would tell me at that location in the genome, transcription factor bound. So I could use next-gen sequencing to query the genome for where this transcription factor binds. Methylation is the same ball game, just different players. There's different places along the genome where there's methylation that has happened. Green dots now are methyl groups instead of proteins. Do the same thing. The chemistry needs to change a little bit because instead of proteins, it's just methyl groups. But the idea is the same. Break up the genome. Only pull out reads that have methyl groups sticking on them. Kick off the methyl group, sequence it, align it back, and then you know where methylation has happened in the genome. That's the idea. I'll leave that with you. There's a blurb of code that shows a package in Bioconductor that is specific to that task. What follows that is a blurb of code for ChIP-seq. There's a package for ChIP-seq. ChIP-seq is transcription factor, finding transcription factors, chromatin immunoprecipitation, flavor, next-gen sequencing. All it does is find where does the transcription factor bind. And there is a package in Bioconductor for that. The rest of this, I think we can move on. The ChIP-seq package does a lot. If you happen to do ChIP-seq stuff, it does do a lot. It's not super easy to use, I don't think. The documentation is a little hard to read. Okay. So I'm going to wrap up with that, the next-gen sequencing stuff. And let's move on to talk about our next topic, which is genome-wide association studies. We're comfortable with DNA, I hope. Contains the genetic code. We're comfortable that within the DNA there are genes. Not everything in the DNA turns into a protein. There are subsections of the DNA that we call genes. Those are expressed and then translated into proteins that do stuff for the organism. There's other locations in the DNA that don't code for proteins, but may code for RNA species that regulate the genome. So DNA and genes. If we think about a gene, like this little ladder that's on the top left here, suppose that's a gene. If we're talking about human beings, then everybody in the room has this gene. If we were to zoom into my DNA at this location, there'd be a sequence of letters that would constitute my version of gene Z. So suppose this is the gene for eye color. Maybe there's one gene that determines what your eye color is going to be. It's your reference manual code, the page in the encyclopedia that says eye color equals. My gene won't look like your gene necessarily. I have green eyes, you have brown eyes. So we will have different letters, same chromosome, same DNA, same location, but different letters. What that means is that, or that explains why there's variation in the population. I have green eyes, you have brown eyes, blue eyes, and everything else. I'm 5'10", you're 6'2", whatever. Variation. We all have the same genes. We're all human beings. So we fundamentally have the same DNA. It's very similar across the board, but there is variation. That's why we look different and everything. Okay, so suppose you wanted to study cancer. What is it at the molecular level or the genetic level that makes some people have prostate cancer and some people not have prostate cancer? At the genetic level, there's going to be the vast majority of the DNA that is the same between people with prostate cancer and not prostate cancer. So in terms of understanding what's going on at a genetic level, it makes sense to limit our attention only to the regions of the DNA that differ. So if we're talking human beings, here's a pile of people with prostate cancer, there's a pile of people without prostate cancer. Suppose that 99% of the genome is exactly the same for everybody. I don't need to look at that portion of the genome. Everybody has to be able to breathe or we wouldn't be living. So the genes that regulate breathing, I guess, I don't even need to look at them because everybody has the same thing going on. There's no variation in those locations. If you want to study the genome, what you care about are locations where there's variation, where there's differences. That's how you're going to explain this person has cancer and that person does not. It's because, or it's associated at least with, different gene at this location. So, one more thing. At this location, the gene for eye color, say, different letters could be here depending on the person, and you tell me the letters, the specific sequence, that will translate to an eye color. So one sequence of letters would be blue, one sequence would be green, and so on. We refer to the different flavors of the same gene as alleles. So maybe you've heard the phrase allele or the term allele. What that means is at our DNA, at this location for the gene, there is a specific sequence, and that sequence corresponds to the outcome, the phenotype, what you would actually see when you look at my eyes. It translates into that. There's genotype, there's phenotype. Genotype is the sequence, phenotype is what we actually see. Okay. So there are different alleles of the same gene. And if you tell me what allele you have of eye color, that tells me what eye color you have. A basic genetic idea, if we were to say, suppose just that one base location we want to look at. So all the human beings in the world, we zoom into this particular location, that specific base for these people. Some of them will have T and A. Some of them will, okay, another thing, it's a little bit complicated. In a human, this that we have on the screen is one portion of a DNA molecule. DNA is 2 strands, and T's go with A's, for example. In a human, there's actually 2 copies of this DNA strand. There's 2 chromosomes, 2 copies of the same DNA. So for me, we could come to my DNA, and here's my first chromosome, and at this blue location, suppose I have TA. Here's my second chromosome. Let's not do that. Let's just say we have T and A at this location. Let's see. What I'm trying to explain is something I bet you've all heard. The idea of 2 copies of the same gene, and they're like what that turns into can depend. Suppose that like this chromosome says blue eyes. This chromosome, which doesn't have to equal the other one, says let's say blue, and this one says brown. Then my reference manual is conflicting. One copy says blue, one copy says brown. So there has to be a tiebreaker. What's it going to be? Is it going to be blue, or is it going to be brown, or is it going to be something else? There's this idea of one of these can be dominant over the other one. So, for example, if it were this case, I have one blue allele, one brown allele. If brown is dominant over blue, I'll have brown eyes, even though I have an allele for blue eyes. So there's dominant, there's recessive. And these speak to how alleles at our 2 copies of the same gene translate into phenotype. Genotype is the ACTG sequence. Phenotype is the actual eye color in this case, what it translates to physically. Genes, alleles, in humans, there's two chromosomes. Stephen, what organism do you usually study? Amazing. And how many chromosomes? 10. Okay. Not everybody has 2 chromosomes. That's called diploid. Humans do, many organisms do, some just have 1. And then there are interesting other organisms that have more than 2, like crops in particular, plants, right, have sometimes wheat, yeah. Bananas, oh yeah. Polyploid, right. So that's another thing that we don't have time to get into in this class, but it is important these days. Most of what we think of classically learning genetics, if we've learned a little bit of genetics, is diploid. We're used to thinking about diploid. There's 2 chromosomes, there's these 2 alleles, there's dominant and recessive, and that's what translates into phenotypes. But then you go to bananas or something, and there's 10 chromosomes, and it's everything's a little different. So just something to be aware of. Not everybody is diploid. All right. So going back to this idea of, we would like to, say, study cancer, but 99-point-something% of the genome is the same among humans, even if they have cancer or don't have cancer. There's a subset of the genome where there's actually differences. And we could get very specific and say, let's only look at the bases, the individual bases that vary in the population. If every single human being in the world has a G and a C at one base, we're not going to learn anything by looking at that, by genotyping that base for everybody. But suppose at this particular location, 60% of the population has T and A, and 40% of the population has G and C. Then that's an informative location. That's a common difference between people. That's going to be called a SNP, single nucleotide polymorphism. We are moving into the topic of what's called Genome-Wide Association Studies, GWAS, and GWAS deals with SNPs, single nucleotide polymorphisms. The idea is, for example, suppose we're studying prostate cancer. Let's go get 1,000 people with prostate cancer, 1,000 people without prostate cancer. For each of the people, suppose we have 1 million SNPs. Human beings, there's millions of bases across the entire genome, and I'll show you when we have time that people have spent a lot of money and a lot of time figuring out of those millions of bases in the human genome, here's the subset of locations where there's common variation. SNPs. Here's 1 million SNPs. So if you want to know what's different between cancer and not cancer, you might as well just look at the SNPs, because that's the only place where there's differences. Genome-wide association studies would start with this case control experiment. This is one way to do it, a case control experiment where you have 1,000 prostate cancer people, 1,000 non-prostate cancer people. Suppose there's 1 million SNPs that have been identified as locations of variation in humans. Then what you would want to do is sequence each of your 2,000 people at those 1 million locations. So here's person number 1. This person has prostate cancer. Let's take a genetic sample from them, a tissue sample or something. And now, instead, I don't want to sequence the whole genome. I don't need that whole thing. What I want to do is specifically go to this base, chromosome 4, location 500,025, and sequence that one base. Is it a T, or is it a C or a G or an A or whatever? This would be a SNP. This single nucleotide location in the genome that, say, takes T and A for 60% of the population, but C and G for 40% of the population. If there's a location in the genome that varies frequently, and I'll define that a little more clearly in a moment, then we call it a SNP. So if we had our 2,000 people, the goal would be genotype each of those people at each of the 1 million SNPs. So roughly speaking, for each of my 2,000 people, I have a column in a data set that tells me somehow what was there. You could just put in your Excel spreadsheet a string, a character, TA or AT, or you could code this numerically. You could say, let's let T and A be 0, let's let C and G be 1 or something like that, in which case you have numbers in your data set. But bottom line, we're going to have some way to query the genotype for each of our individuals at each of the SNPs. And then what would you want to do? You'd want to ask which of those SNPs are associated with disease status. 1,000 people with prostate cancer, 1,000 people without prostate cancer. We're looking at 1 million SNPs. I really only care about the subset of those SNPs where there's statistically significantly different profiles, allelic or genotype profiles at these SNPs. So for this particular SNP, the one that we have boxed in blue here, suppose we have 1,000 prostate cancer, 1,000 non-prostate cancer. We go in and we genotype that SNP for everybody, and the majority of the cancer people had T and A. The majority of the healthy people had C and G, maybe. And if we could put a p-value on that comparison, 60% with this genotype versus 40% with the other genotype, is that statistically significantly different? Do a test. You could do a test of proportions, like a 2-sample test of proportions. What's your regression model alternative? What kind of regression could you do with genome-wide association studies? Cancer versus normal. Here's a SNP. Is it statistically significantly associated with cancer versus normal status? You could do a non-parametric something, I'm sure. What kind of regression do you use when you have a binary response? Logistic regression. Cancer, no cancer, yes or no. Let's model that in terms of genotype. Genotype, or SNP X, this one right here. Let's do a logistic regression model, cancer, yes or no, modeled in terms of intercept plus genotype effect. Put a p-value on that genotype effect, and then we're back in the context like we were doing with FDRs earlier. I have millions of features, or 1 million features in this case. Each one is a SNP. For each one of them, I have a p-value. Turn those into FDRs. You have a list, a subset of SNPs that are statistically significantly associated with cancer. So just to clarify that, suppose our response is 1 if an individual, so let's say yi, is 1 if individual I has cancer, and 0 otherwise. And for, let's say, the SNP that we were looking at up above, let's say xi, my covariate, is equal to, let's say, 1 if the allele corresponds to, let's say, phenotype, let's do it this way. If, let's say there's only 2 alleles. You're either brown or you're blue. To be brown, you have to have 2 brown alleles. I'm sorry, to be brown, you have to have just 1 brown allele, because brown is dominant. To be blue, you have to have 2 blue alleles. So you're either brown or blue. This would be brown. This would be brown. This would be blue. You are either brown or blue. I could do a test. When we get our sequencing data, we have 2,000 rows in our data set. And for this SNP, we have a bunch of ones and zeros. So in my data set, 2,000 rows and a column for this SNP in which there's just zeros and ones. If I wanted to ask, so our data might look like, we'll put individual in the first column, just 1, 2, ... 2,000. Let's go ahead and put group condition. So we'll say the first 1,000 are cancer, and the last 1,000 are healthy. And then we're going to have a column for each of our SNPs. SNP1, ... SNP, let's say 1,000,000, 1e to the 6. For SNP1, suppose that's the one that we outlined in blue up above, we're going to have a bunch of zeros and ones. So yes, they had the one phenotype, they had the other one that time, this one had the one phenotype, and so on. If I wanted to assess SNP1, is it associated with cancer status? I could do a couple of things. I could do a simple 2-sample test of equality of proportions, like the proportion in the cancer group who had phenotype 1 for SNP1 minus that proportion in the healthy group for SNP1 divided by some standard error, like a t-test, t-statistic. Get a p-value, good deal. The other thing that I was just mentioning is logistic regression, which is just like this, but it's an extension or a generalization of doing a t-test. So with logistic regression, we would say the log odds, the log probability of cancer for individual I divided by 1 minus the probability for cancer. That's an odds. P over 1 minus P is an odds. Log transform the odds, and you're doing logistic regression. So what we would say is that that response is equal to an intercept plus a, quote, slope, although it's not really, that doesn't make sense to think of it as a slope since x is binary. It's an effect times, let's say, x, let's make this x1i, as in for SNP1. SNP1, individual I. That's the logistic regression model, and the p-value that we would have gotten from a 2-sample test of proportions is going to be analogous to the p-value we'd get for testing is beta 1 equal to 0 or not. If beta 1 is equal to 0, then SNP1 is not associated with cancer status. If it is, if it's not equal to 0, there is an association. If it is equal to 0, there's not evidence of an association. So all I would need is a p-value for beta 1. The nice thing about thinking of this problem in terms of a regression model, logistic regression, is that suppose you had, in addition to these variables over here, suppose you had a column for gender and a column for age and race and stuff like that. All you need to do now is add in covariates. So gender and so on. In which case, you're asking, is there an association of SNP1 to cancer adjusted for covariates, which is always a good idea to limit the possibilities of confounding. So when we do GWAS, one major flavor of GWAS is this. You have millions or hundreds of thousands of SNPs. We want to know which of them are associated with some usually disease status or some condition. That could be a logistic regression problem. Another twist on that is if instead of cancer versus normal, yes or no, 0, 1, if instead we have a continuous response that we're interested in, like PSA, maybe prostate-specific antigen, a protein that your doctor will quantify, as a risk factor for prostate cancer. Suppose we wanted to ask, is PSA associated with SNP1? Does SNP1 associate with PSA? That's not a logistic regression problem anymore because PSA level is going to be a numerical value. I don't know what the usual range is, but your PSA is 62.4. It's not 0, 1. What would you do? Sorry? You could categorize it. Do you have to? Okay, so suppose you have a continuous number. You could always categorize your continuous number and do categorical data analysis. Generally, just do linear regression is an option. If you categorize, you will lose information, generally speaking. If you have numerical continuous data, it's a fair move to categorize it. Sometimes that's good, sometimes it's bad. It's good if your data are really noisy, maybe, and it's too noisy. Break it into categories simplifies the problem. But generally speaking, there's more information in the actual numbers than there is in categories. So a natural thing to do would be, instead of logistic regression, let's just do linear regression. So if you have a quantitative response, you're going to do this again, but it's not logistic regression anymore. It's linear regression. So we might say if yi is prostate-specific antigen level for individual I, we could say regression model. Intercept, SNP1 effect, gender, age. And the same goal holds. Let's do inference on beta 1. Is SNP1 genotype associated with PSA, adjusted for covariates? All right, that's my intro, and I was going to spend a little time, we've got 10 more minutes today, looking at an article. And I'll let you guys, I recommend that you read this article, maybe before Friday if you have time. Bush_12 is an overview paper of GWAS. All right, so this is a PLOS computational biology paper. It's a good one, I think, as a kind of a survey of what GWAS is all about. All right, one thing that I have not said yet, I'm just going to kind of scroll through the paper and comment on a few things that come up. One thing that I did not say, okay, so SNPs, we have said, are single locations where there is variation. And I mentioned this, but I didn't emphasize it. It will be called, this location will be called a SNP if there's variation and if the variation is, quote, common. What does that have to do with anything? Some diseases, like cystic fibrosis, are caused by rare variation at a specific location. But that location is not called a SNP traditionally. So what's the difference? What makes this location, say, a SNP, and another location that's just like it that causes something like cystic fibrosis? Why would that be called something else, e.g. mutation? Cystic fibrosis has a location where there's differences, there's variation in the population, and it's called a mutation. We're not going to be looking at mutations in GWAS. A mutation is a very rare change that has a big effect. Let me show you a picture over here. Think about genetic changes. What happens if I smoke lots of cigarettes and I get a mutation in a location in my genome? That may do nothing, or it may kill me, depending on where in the genome it is. A change in my genome, maybe one specific base. Suppose that one specific base gets changed. It's possible that that's a really bad place to have a change in your base, in your genome, in which case it has a big effect. The paper refers to it as highly penetrant, as in that one change gave you a disease. Those are pretty rare. That we don't open up the CNN and find headline being "the cancer gene has been found." There is no cancer gene. There is a cystic fibrosis gene, and there are some diseases that can be explained in a simple way. One mutation will do it. Cystic fibrosis, there is what we could call a SNP. There's a location where there's variation, and it causes cystic fibrosis. It's not called SNP because that variation, that change, the mutation, is actually really rare. What GWAS is about is trying to study different types of relationships. If you're looking for the magic gene that causes a rare disease, there are different techniques that you would use. I'll mention it next time. Family studies, family association studies. Cystic fibrosis, if you have this mutation, you have cystic fibrosis. It's sufficient to go look at a family, parents, children, grandchildren, and so on. Let's watch this family and genotype the heck out of them and see who has cystic fibrosis, who doesn't. If you do that, you can quickly find the magic gene just by looking at a family that has cystic fibrosis people in it. But what about prostate cancer? My dad had prostate cancer, but you would not learn much by genotyping him and genotyping me, because prostate cancer is common in the population, and it's thought to be driven by many small changes. Highly penetrant cystic fibrosis, think one major mutation. One mutation, very rare, that has a big effect. Most of the more interesting, the more common diseases, like prostate cancer, cancer, cardiovascular disease, are thought not to be driven by one magic gene, but rather driven by combinations of many small changes. That's why we care about this SNP idea, because the scientific community has become convinced that many of the more common diseases, in order to understand them at a genetic level, we're going to need to go find many locations of changes. So if we want to find the locations of changes, might as well just look at locations where we know there are changes. And if we're looking at common diseases, let's look at places where there are commonly changes. Not a very rare mutation, but locations where, for example, I have green eyes, you have blue eyes. And everybody has variation there, lots of variation there. GWAS is located conceptually, or it's targeting conceptually, common diseases that are driven by common variations that individually have very small effects, but collectively result in cancer or disease. Okay. Section 2 mentions Snip, single nucleotide polymorphism. If you want to read some more detail about what SNPs are, they are by far the most abundant form of genetic variation in the human genome. So SNPs are kind of the basic unit of observation, arguably now. Okay. SNPs, we will talk in terms of allele frequencies. This is how we are specific about, is it a common or a rare change? We're going to call a location where there's variation a SNP if it is not too rare. Basically, that's it. Cystic fibrosis mutation, I don't know what the percentage is, but some really 1 in 10,000 people or something are expected to have that mutation. We are going to be concerned with locations that vary. I don't know what's the convention. 5%. Okay. So apparently a common convention nowadays is, let's call a location in the genome a SNP if its minor allele frequency, as in the less frequent of the possibilities, is at least 5%. So that's common enough, is what the idea is. Okay. This section right here, 2.2, talks about the issue that I was saying. Cystic fibrosis is different from prostate cancer. Cystic fibrosis, high penetrance, rare mutation, in which case you can apply a different technique called linkage analysis. It's a family-based study. Just look at a family that has cystic fibrosis, sequence everybody, eventually you'll find the location where the mutation happened. So GWAS is targeted at not that. Are they referring to for this? Because usually when you do SNPs, if your SNP is it's not viable, you should get rid of it. So are they referring to just the minor allele frequency too low, or are they referring to, like, instead of it just being A's and T's, there's a G in there somewhere? Because the SNP should all just be A's and T's or ATX. So you know what they're talking about? I think it varies depending on the organism you're studying, depending on, is it diploid or not. In the diploid case, I think it is just major, minor. Two possibilities, and if the minor one is not too small. I don't know what the convention is for, say, 10 chromosome organism. Well, I just know that it's hard to do GWAS if you have more than 5 alleles. So if you have, at SNP, you have 3 different alleles, you have A, T, G, instead of just A and T across all of the individuals, then it's really hard to do anything because you can't estimate the effects. Because most of the programs are set for just 2. Okay. Most of the programs only can handle 5 alleles. So the comment here was that everything we've been saying is there's 2 possibilities for my SNP. You're either 1 or you're 0. There are contexts where there's more than 2 possibilities. In principle, that does not present any issues because you can always do regression in that case still. That said, software may limit you. If there's a commercial software package for GWAS, it may only, like you said, be set up to deal with the 2-class case. But if you had, let's say, 3 possibilities, you could still think of it as a regression model. You would just have 1 extra coefficient in your model. So you'd have like a beta 0 plus a beta 1 x let's do it this way. Indicator variable that x1i equals 1 plus beta 2 indicator variable that x1i equals 2. Let's leave that off. What that means is that beta 0 is the response for x equals 0. Beta 1 is the mean change comparing x equal to 1 to x equal to 0. And beta 2 is the mean change comparing x equal to 2 to x equal to 0. So you've got everything covered in there, and you can get separate p-values and everything. You can do it. So I would say if you're in that boat, learn how to do the regression yourself, because the package, like the Snip association package in Bioconductor, all it's doing is linear regression, logistic regression. So if I were doing GWAS, I probably wouldn't even use the package. I would just do regression. That's all it's doing. And if you're going to do regression, you've got complete control over, is it 2 groups or is it 3 or whatever. So you could come into R with your data with more than 2 possibilities and do GWAS, get p-values and FDRs and everything by just doing regression. And I can help with that if you want to talk later. We are out of time. I'm going to go just a little bit more through this. I wanted to mention a couple of things in this paper, and then I have some R code with a bioconductor package that has GWAS stuff in it. We need to talk about this idea of linkage disequilibrium and an idea of Hardy-Weinberg equilibrium. So I think, yeah, I've got a fair amount more to talk about with GWAS stuff, and then we'll do some examples, obviously. Okay, homework's due tonight. Other questions? All right, I'll see you Friday.